Thank you for this kind introduction. First of all, I would like to thank HORIBA Scientific for the opportunity to present a part of my research activities conducted in the Laboratory of Pharmaceutical and Analytical Chemistry of the University of Liège. The topic of this webinar will be focused on imaging and more especially on Raman Hyperspectral imaging. On the next half hour, I will demonstrate through case studies that Raman imaging can be considered as an essential tool in the pharmaceutical and in the biomedical field. The outline of the presentation will be based on 4 parts. The first 2 parts will allow to set the scene of hyperspectral imaging and fundamental concepts of Raman spectroscopy, instrumentation, and data analysis. The third part will be dedicated to pharmaceutical and biomedical application in order to illustrate the power and the suitability of this technique. Finally, I will end the presentation by drawing some conclusions. Raman hyperspectral imaging is based on the combination of 2 techniques, namely Raman spectroscopy and imaging. This combination allows to acquire simultaneously spectral and spatial information from a sample. The identification, solid-state characteristic, and quantification of sample components are performed on spectral data, while the distributional homogeneity of components, structure of the pharmaceutical solid forms, particle size, and coating thickness are based on spatial data. In 1928, it was observed by Sir Raman and Krishnan a shift in the wavelength of a very small part of the radiation scattered off from molecules exposed to a monochromatic light. The discovery of this phenomenon of an elastic scattering of light, known as Raman effect, was awarded in 1930 with the Nobel Prize in Physics. It is associated with a change in the polarizability and vibrational excitation of molecules. As illustrated on this slide, 2 phenomena can occur: Rayleigh scattering, which is non-informative and filtered out, and Raman scattering, which is informative on the molecule. The Raman scattering is designated as Stokes-Raman scattering when the photons are scattered with a lower energy after the interaction with the molecule. In the last decades, Raman spectroscopy emerged as one of the most powerful techniques in analytical sciences due to its numerous advantages, such as its high molecular specificity, its ease of implementation, and its non-destructivity. A Raman spectrum is very informative and can be considered as a fingerprint or the ID card of a molecule. The band intensity is related to the polarizability, the orientation, and the detectability of a molecule. The band position depends on the chemical structure. The band position shift gives information about that information and the stress. And finally, the band width is related to the solid state of the molecule. As said previously, Raman imaging is an analytical technique enabling to acquire spectral and spatial information at the same time, based on a working area defined by the analyst and according to the objective of the technique. Therefore, it is possible to observe the spatial distribution of molecules within the samples through the spatial information, while their identification and quantification are based on the spectral information. As shown on the right side of the slide, data are collected in form as 3-dimensional data, x, y, and lambda, with x and y as spatial dimension and lambda as spectral dimension. Three imaging modes are possible to acquire an hyperspectral image: point mapping, light scanning, and global imaging. Regarding the point mapping mode, the analyst begins by drawing a grid or mapping area, representing the spatial position on the sample surface and defining the step size, which is the size between 2 spatial positions. When the analysis starts, spectrum at a specific spatial localization is acquired, then the sample moves to the following position for new acquisition, and so on until the whole grid is covered. This mode is the most frequently used due to its high flexibility and the high spectral quality in terms of resolution and spectral range. However, the analysis time can be very long, depending on the spatial resolution, the grid size, and the acquisition time of one spectrum. For the line scanning mode, as with point mapping mode, spectra are acquired according to a defined spatial position, but in this case, spectra of whole lines of the mapping area are recorded simultaneously. It presents the same advantages as point mapping, while decreasing the analysis time. For the last one, global imaging mode, the entire surface is illuminated and the whole mapping area is acquired simultaneously at a single wavelength. Tunable filters move to select another wavelength, and so on until the whole spectral range is recorded. Therefore, a global imaging configuration allows to record the whole map with a fast acquisition time. However, the spectral and spatial resolution are limited respectively by the kind of tunable filters used and the pixel size of the detector. The first key parameter of Raman imaging is the spatial resolution. The spatial resolution represents the focus diameter of the light spot arriving on the sample. It is a function of the laser wavelength, lambda, and the numerical aperture, Na, of the objective, as exposed in this equation. To improve the spatial resolution, lambda must be as low as possible and Na as high as possible, inducing a little light spot on the sample and thus a little analyzed sample area. For example, using a laser wavelength of 532 nanometers and an objective with a Na equal to 0.9, the theoretical spatial resolution is equal to 361 nanometers. However, the spatial resolution is close to 1 micron, due to the optical behavior with Raman microscopy, which is much more complex. Another important point to be highlighted is the notion of confocality. The confocality notion appears with the use of confocal Raman microspectrometers. The term confocal is defined as having the same focus, meaning that the data acquisition of the sample is performed by measurements taken at the same focal plane. As shown on this slide, a first pinhole or slit is present in the front of the light source. It focalizes the light on the sample, increasing the image quality by reducing the scattered light. The second pinhole, which represents the detector confocal pinhole, has two advantages. First, it rejects the scattered radiation coming from an out-of-focus point, and secondly, it permits to realize images in the depth plane of the sample, allowing analyzing the different layers on this later. It is interesting to use a confocality for pharmaceutical analysis. Indeed, it is possible to acquire information in the z-plane and thus to obtain three-dimensional spatial information of the sample. Moreover, it is also possible to perform optical slicing of the sample without the use of successive preparation steps. Raman Hyperspectral imaging has numerous advantages, among which the following are noteworthy. This analytical technique enables to acquire spectral and spatial information at the same time. It is a green and non-destructive technique having a fast data acquisition. A Raman spectrum contains sharp spectral bands and is sensitive to the solid-state characteristic. Compared to infrared, the presence of water does not hinder its applicability, because water is a weak Raman scatterer. And the last one, the examination of a wide diversity of samples, including semi-solid, mixture, liquid, or suspension, is possible with a minimal sample preparation step. Regarding the limitations, an issue for the applicability of Raman spectroscopy is the fluorescence and the sample degradation. However, several Raman laser sources from UV to NIR region are available, and a trade-off must be done for choosing the most appropriate one. By selecting a NIR region Raman source, the fluorescence of the samples can be significantly reduced. Even if Raman spectrum contains sharp bands, multivariate data analysis is most of the time required to extract the relevant information from complex or noisy signal coming from the sample. The spatial resolution is close to 1 micron. It is also important to keep in mind the weak sensitivity of classical Raman scattering, but the use of other techniques such as surface-enhanced Raman scattering is possible to circumvent this limitation. The last one is the total analysis time from several minutes to hours, depending on the site of the working area, the step size, the nature of the analyte, and the complexity of the matrix. Once acquired, hyperspectral data must be analyzed to extract the information sought. Hyperspectral data analysis may be divided in 3 main parts: pre-processing of raw spectral data, processing of pre-processed data, and post-processing of the image. The pre-processing step aims at correcting the perturbation that occur during the analysis or limiting the effect of undesired phenomenon to facilitate the access to the relevant information. Regarding the pre-processing of pre-processed data, univariate or multivariate data analysis can be used depending on the data complexity. When a specific peak corresponding to the interest compound may be found, without any interfering adjacent peaks related to the other compound, the easiest way to analyze the data is the extraction of the distribution map at the corresponding wavelength. In the other cases, it is necessary to use multivariate data analysis approach based on exploration, resolution regression, or segmentation techniques. Each technique has its own specificities and will be used depending on the available and the desired results. More and more user-friendly software are provided directly by the manufacturer, such as HORIBA with LabSpec, in order to help the analyst to manage conventional sample analysis. On this slide, 2 examples of pre-processing are illustrated: spike correction from left to right and baseline correction from up to down. Raman spectra and more generally spectra acquired with a charge-coupled device CCD detector are likely to be contaminated by cosmic rays. These highly energetic subatomic particles from space may hit a CCD pixel and saturate it, providing a single, unusually high intensity value. When very few spikes are present, a manual correction may be envisaged. Some algorithm has been developed if manual correction is not possible, or if an automation of the correction is needed. When performing spectral analysis of a sample, drift in the baseline may occur due to fluorescence. Therefore, it is sometimes important to remove this perturbation using different approaches such as asymmetric least square, mixture model, or weight least squares and polynomial filter. In the most applications presented during this webinar, hyperspectral data cubes were acquired using a Labram HR evolution, equipped with a 2-dimensional MCCD detector and a laser excitation wavelength equal to 785 nanometers. This wavelength is a good compromise between signal intensity and fluorescence risk. A 50x long working distance objective was used, and a grating with 300 grooves per millimeters was selected to acquire a large spectral range in one shot. The confocal wall was fixed at 200 micrometers, and the acquisition of the data was performed using the point mapping mode. The first conventional application relates to identification and the distribution at a microscopic scale of A components in a solid dosage form as shown on this slide. Hyperspectral data cube was obtained from a commercial tablet and was analyzed using multivariate curve resolution alternating least square. MCA-RLS is a technique of choice when dealing with samples of unknown composition, taking into account its intents the recovery of pure spectra of the chemical constituent of an unresolved mixture when no information is available about their nature and the composition of these mixtures. The use of a reference spectral database has allowed to identify each result spectrum obtained with MCA-RLS. The relevant correlation coefficients are highlighted on the figure on the right. A visual inspection confirms the good agreement between the result and the reference spectrum. Finally, the distribution of each component was determined thanks to the spatial information. On this side, one can visualize the distribution of 8 components in the tablet, which seems to be homogeneous. From left to right, sodium bicarbonate, sodium saccharinate, paracetamol, and lactose, and also ascorbic, acetylsalicylic, citric acid, and sorbitol. In the framework of the detection of counterfeit medicines, Raman Hyperspectral imaging combined with chemometrics such as MCA-RLS, as shown previously, or ICA proves to be a suitable technique. It allows to identify main components of a suspected counterfeit. The result of a case study dealing with suspected counterfeit killing tablets are summarized on this slide. In the first step, some tablets were analyzed by HPLC. No API was detected, but no relevant information regarding the composition of the tablet was obtained. Facing this lack of information, the tablets were analyzed by Raman imaging, and the result spectra were compared with the reference spectra of the database. This comparison study allowed to confirm the HPLC results, no killing detected, and also allowed to identify metronidazole as a potential candidate compound present in the suspected tablets. This result was confirmed by additional investigations based on mass spectrometry and nuclear magnetic resonance. Depending on the level detail for the spatial distribution required, the analysis can be performed from macro to microscopic scale. The level detail of the image will be led by the laser spot size and by the step size of the working grid, which corresponds to the distance between 2 neighboring analyzed pixels. Of course, a balance must be struck between the step size and the size of the analyzed area according to the total analysis time needed to acquire hyperspectral data. In order to illustrate this point, 3 maps of the same tablet containing sodium bicarbonate, citric, acetylsalicylic, ascorbic acid, sorbitol, and paracetamol will be presented. For the first one, the size of the map is equal to 4 by 4 millimeters with a step size of 20 microns. As you can notice, a good survey of the sample is obtained. If required, the level detail of the image can be improved by decreasing the step size and also the size of the working area in order to keep a reasonable acquisition time. The next image will represent the area within the wide square. The size of the map is now equal to 5 microns by 5 microns with a step size of 2.5 microns. As one can see, a lot of details regarding the size, the shape, and the spatial distribution of the particles can be obtained. For the last map, the size is equal now by 100 by 100 microns with a step size of 1 micron, allowing to focus the analysis on one particle and its surrounding area. Thanks to the spatial distribution at microscopic scale of the main components of a solid dosage form, this technique can also be used to determine the influence of formulation on the spatial distribution of the API. As shown on this slide, the comparison between two formulations, 1 and 2, containing the same API colored in blue, is easy and obvious. The structure of both formulations is not the same. As previously said in the introduction, the Raman spectrum of a compound is sensitive to its solid state, allowing to use Raman imaging in polymorphism study. Indeed, distinct crystalline forms commonly exhibit differences in relevant properties in terms of bioavailability, solubility, rate of absorption, stability, and processability. As an example here, polymorph characterization of mannitol which exists in 3 forms is shown. Beta, which is usually used in pharmaceutical products, and alpha and gamma, considered as contaminant. Based on a visual inspection of Raman spectra, a clear spectral differences can be highlighted between polymorphs. Taking into account the spatial information, the location of each polymorph can be directly determined in pharmaceutical products. In addition, the overlay of each map allows to build a global image of the sample in which the spatial distribution of each polymorph can be compared, as presented on the figure on the right side. Alpha mannitol in blue, beta in red, and gamma in green. Another key aspect of a pharmaceutical product that can be studied by Raman imaging is the particle size of active pharmaceutical ingredients. The particle size on the formation of agglomerates during the manufacturing process are critical with poorly water-soluble drugs. Image processing and analysis obtained from a map allow to calculate several parameters such as the number, the modal, minimal, and maximal diameter of the particles. Thanks to the confocality of Raman microscopy, the estimation of the particle size in 3D, X, Y, and Z is also possible as illustrated on this slide. In order to obtain this 3D graphic representation, successive optical slices were performed based on a depth profile. Besides the investigation of the particle size, another interesting application of Raman imaging is the determination of the coating or membrane thickness in the framework of quality control or sustained release systems such as implant, pellets, and tablets. Indeed, this thickness has a dramatic influence on the rate of drug delivery. The accuracy of the results will depend on the step size of the map and the laser spot size. On the left side, one can see 3D and 2D images of a transversal cut of an implant. Regarding the 2D composite image, the inner part of the implant is shown in green and the membrane is shown in red. Its thickness was evaluated around 60 microns. On the right side, visible and Raman images of a transversal cut of a pellet are presented. The coating is also shown in red. Based on these images, the investigation of control release systems is straightforward. As Raman spectroscopy, Raman hyperspectral imaging can be used in quantitative application in which a global or single termination could be an issue. A typical example is scored tablets. Each half of the scored tablet has to contain the same amount of API. The development of the quantitative application based on Raman imaging is close to Raman spectroscopy. Calibration mapping and reference methods are used to build a regression model such as PLS regression. In this example, calibration mapping from 50 to 150% of the target concentration were required, and an HPLC was used as reference method. A good agreement between the percentage of API measured by the reference method and the percentage of API predicted by Raman imaging was obtained, as one can see on this slide. Its analytical performances were determined based on a total error and risk-based approach using the accuracy profile as decision tool. The quantitative regression PLS model has been fully validated within the plus-minus 10% acceptance limits. The classical plus-minus 5% acceptance limits for the assay of pharmaceutical product were not achievable because of sampling issues. Only a small slice of a sample is mapped, and the representativity of the sample is directly linked to its homogeneity. But the main advantage of using this technique is to obtain reliable quantitative information at the pixel level instead of a global information of the sample as presented on the map, with an X-min and X-max value equal to 88% and 116% respectively, and the mean value equal to 103%. As said in the introduction, the detection of low-dose components such as impurities is not possible due to the weak sensitivity of Raman spectroscopy. One way to circumvent this limitation is surface and end-Raman scattering, which enables to dramatically increase the Raman scattering of molecules which are absorbed or very close to metallic surfaces. Gold and silver nanoparticles are mainly used as source substrates. As classical Raman scattering, source enables to obtain spectral information of a very low dose components, which allow qualitative and quantitative analysis. In this example, source imaging was developed to quantify 4-aminophenol, which is the main impurity of paracetamol, assessing a limit quantification below its limit of specification of 1000 ppm. Citrate-reducible nanoparticles were used as source substrate. As one can see, a quantitative approach was successfully developed, enabling to quantify 4-aminophenol from 0.025 to 0.2% in paracetamol tablets. Considering the development of a quantitative application, the optimization of the way used to cover the sample is a critical step in order to obtain an homogeneous covering of the sample surface and thus repeatable results. Besides pharmaceutical applications, Raman imaging is also an interesting and suitable technique to investigate biological samples. Applied to biological samples, the same kind of information can be obtained. However, the interpretation of the results in terms of spectral data and image is not straightforward due to the biological nature of the sample. In addition, keen pre-processing and chemometric tools are very often required to do the extraction of the relevant information from the hyperspectral data cube. Moreover, the lack of sensitivity of Raman imaging can be an issue, and thus source approach has to be investigated. The first biological application presented today is dealing with the analysis of urolithiasis. Chemical composition is not the only point to take into account when analyzing this kind of sample. Most of the time, calculi are made of more than a single element, and the succession organization of the different chemical elements also gives clues on the stone growth history. Therefore, the use of Raman imaging enables the possibility to directly link stone composition and stone morphology in a single analysis, as illustrated on this slide. As can be seen, uric acid in red, uric acid monohydrate in green, and calcium oxalate monohydrate in blue are present in the stone and form several successive layers. The second biological application concerns the determination of the chemical composition of giant feeding cell walls of poplar. In this study, a focus was made on the identification and spatial distribution of pectin in green, lignin in blue, and cellulose in red, which are the three main cell wall components that are well characterized by Raman imaging. To conclude, I am convinced that all examples presented today have allowed to demonstrate that Raman Hyperspectral imaging can be considered as an essential tool in the pharmaceutical and biomedical field. Indeed, hyperspectral data cube combines spatial information at a microscale and spectral information allowing identification to quantification of pharmaceutical and biological samples in a non-destructive way, requiring no little sample preparation step, and compatible with moisture and water media. Finally, I would like to thank each scientific team of the Center for Interdisciplinary Research on Medicines, which collaborate on this topic: the Laboratory of Pharmaceutical and Medical Chemistry, the Laboratory of Pharmaceutical Technology, the Department of Clinical Chemistry. I would like also to thank le Laboratoire de Biologie Végétale de l'Université Libre de Bruxelles, SMB Tech and Galifar MF for the support, and HORIBA Scientific for the opportunity to present a part of my research activities. Of course, I would like to thank you for your attention.